Welcome to The New Chemist. We're glad you're listening. Feel free to download this podcast on Apple Podcasts, Google Podcasts, and Spotify. Here on The New Chemist, we discuss chemistry, which simply put is the science of change, as well as careers, community, research, and COVID-19. We're happy you're tuning in. My guest today is Dr. Robert Langer. Thanks for joining me today. It is good to hear from you. Just briefly, I'll inform my audience about you. Dr. Robert Samuel Langer, Jr. is an American chemical engineer, scientist, entrepreneur, inventor, and one of the 12 institute professors at the Massachusetts Institute of Technology. He was formerly the Germershausen Professor of Chemical and Biomedical Engineering and maintains activity in the Department of Chemical Engineering and the Department of Biological Engineering at MIT. He is also a faculty member of the Harvard-MIT Program in Health Sciences and Technology and the Koch Institute for Integrative Cancer Research. In 2015, Dr. Robert Langer was awarded the Queen Elizabeth Prize for Engineering. He has numerous accolades and achievements. Please welcome Dr. Robert Langer. Okay, Dr. Langer, thank you so much for joining me today. It is good to have you here.
My pleasure.
Yes. So what has been some of the most beneficial advice you have received?
Well, I would say that, it's not so much advice as just a role model. Judah Folkman was my postdoctoral advisor, and he was the kind of person that believed anything was possible. I don't know if he used those words, but he lived that, and so I think that thinking that anything is possible, and I think recognizing when you think that way, you're going to often get criticism. There's going to be a lot of roadblocks, and then not giving up. And I think, he never said it quite that way, but that's kind of what I learned from him as a mentor, to dream big dreams and never give up.
Oh, wow, that's very good. It's very good. Persistence is important. So, do you have any advice for those wanting to pursue the field you are currently working in?
Well, what I just said is part of the advice. I think it's important to do things that are transformative and not incremental, but recognizing that if you do that, you will run into roadblocks and you will fail sometimes. So, so the important thing is, as you just said, is persistence. Persistence is very important.
Yeah, that's true. Dr. Langer, I listened to several of your interviews, and one of the things that stands out to me is that your academic path, your academic journey was not easy. You encountered some challenges, and I think it's very commendable that you were able to persist and be successful amidst those challenges. So, in line with your major, why did you choose chemical engineering as a field to major in?
Yeah, I would like to tell you that it was really well thought out, but I can't say that. Initially, when I was in high school, my dad and my guidance counselor said, well, you're good in science and math, so you should become an engineer. So I became an engineer. And, and then when I was at Cornell as an undergraduate, the course I liked the best and the one I was best at was chemistry. So I became a chemical engineer.
Okay. Oh, wow. Wow. So
I'm glad I did. I think it's been a great profession.
Okay. Yeah, I would say so too. So in terms of your doctoral studies, is it along the same lines that you chose chemical engineering to do your doctoral studies in, or are those things the deciding factors, the things that affected you in your undergraduate years?
Well, so I for a long time, I didn't have a very clear idea of what I wanted to do. As an undergraduate, like I say, I majored in chemical engineering for the reasons that I said. As a graduate student, in a way, it was just a continuation of that. I still was kind of searching for something I wanted to do. And when I was a graduate student, I got involved in a lot of other things too, like helping start a school for poor high school students and things like that. And developing new chemistry and math curricula. And so it wasn't until my postdoctoral work that I really sort of found a research direction, and that was by being in the hospital. I worked in Children's Hospital as a postdoc, and I was really the only engineer there. And as I mentioned, the man I work with, Judah Folkman, he was a great role model, and I started to think about how I could solve medical problems with my chemical engineering background.
Okay, yeah, that's very good. So you mentioned how you started the school, and I remember hearing about the school in a previous interview. So my question to you is, why and how did you go about starting that school?
Well, actually, so my interest in teaching started when I was at Cornell because I was a teaching assistant, and I really loved that. And so when I came to Boston the next year and Cambridge to join MIT as a student, graduate student, I got involved in doing some tutoring activities in Roxbury and Cambridge. And so there were some people, Peter Haggerty, Neil Didrikson, Paula Didrikson, they were interested in starting this school for working class children, for kids. And they had heard about me, so they asked would I help on developing the math and science programs. And I thought this sounded like a very worthwhile thing, so I did.
Oh, that's very good. So how have you maintained vision and teamwork in your environment? How have you maintained those things in your work environment, in your lab? How do you maintain those things?
Well, I guess there are different things. So vision, I try to encourage everybody in the lab to think big and to dream big dreams and to do things that can hopefully have a big effect on the health and well-being of the world. And so, in terms of teamwork, and I encourage everybody to work together. We have a lot of people. We have over 100 people in the lab. But I try to hire, this may sound almost silly, but I try to hire very nice people who get along with each other, but who also really want to make a difference in the world. And so that's what I've tried to do repeatedly. So
Yeah.
and they've been great. The people we've had in our lab have been outstanding.
Yeah, that's very true. So in terms of your success, I think by any standard of measurement, international, national, I think people can all agree that you have been successful as a pioneer in the field. So what would you say has complemented the most to your success? Would it be mentoring? Would it be networking? Would it be your approach to studying or learning or teaching? What would you say has been the main driver or impetus in which you have been successful?
Well, I think to the extent that I have been successful, and that may be debatable, but to the extent that I have, I really think it's been the people. I've had wonderful, wonderful people working in the lab, and they've been great. I like to think that they feel like I've treated them well and that I've encouraged them to live their dreams. But really it's having just tremendous people. MIT's been a great place, and we've just had a tremendous number of very, very good students and postdocs in the lab. And, by any measures, I think 19 are now in the National Academy of Engineering. I think 16 in the National Academy of Medicine, and they've won all kinds of awards, started companies. They've been very, very successful. So to me, it's been them.
Yeah, it's true. People are important, very important. So given all your responsibilities and accomplishments, how do you strive to maintain a balanced life?
Well, my wife is very good at making sure I do. When my children were little, she told me that if I wasn't traveling and that I shouldn't, that I needed to be home by 7 every night to spend time with the kids. And when I traveled, of course, right now I'm not traveling, but when I did travel, certainly when the children were young, or even at home, I would never be gone for very long. I kind of learned that was important to them. So just to give an example, 5 years in a row, I had to go to Israel for something like a lecture, an award, or a degree, honorary degree. And all those 5 times in a row, 5 different years, I didn't use a hotel. I just flew over, I spent part of the day there, and then I flew right back.
Wow.
They have a 1 a.m. flight from Tel Aviv to New York, so I was able to do that. But anyhow, so I never would be gone for very long.
Okay, wow, yeah, family is a very important thing, and so you would say your family is the basis on which you've been able to keep a balanced life.
Well, that and my wife has been very good, Laura. She tells me what she thinks. So if I did something, if I was probably spending too much time doing other things, she would certainly let me know.
Okay, that's good. So, in terms of your impactful ideas, what would you say have been your most impactful and effective ideas to date?
Well, I think there's been 3 or 4 areas that's hard to, that have been important, I guess. One has been the work we did with Judah Folkman on isolating the first angiogenesis inhibitors, the first blood vessel inhibitors, and that's led because of work that others had done after that, at companies and so forth, to many new drugs that would be useful in treating cancer and eye diseases. The second is probably the work on new materials and controlled drug delivery, which also, and that includes nanotechnology, and that's also led to quite a number of products by different groups again. And finally would be the idea of with Jay Vacanti of tissue engineering, where you could combine materials and cells and make new tissues and organs, whether that be a organ on a chip or a new tissue in the body like artificial skin.
Okay, okay. So, in terms of you finding and seeking the right environment for you to thrive scientifically and intellectually, how did you go about finding that? What was your guide, or what was your rubric or basis on which you found that environment? I take it was at MIT, but how did you find that?
Yeah, well, I mean, so there are different ways. I mean, I really, I suppose the experience I had at Children's Hospital, that was really key. And then MIT, I mean, that was, I guess, good luck. That they hired me, and things worked out there. Ultimately, though, you create your own environment. Really, when you run a lab,
Okay.
it's the environment you create for others. That ends up making a difference.
Yeah, it's interesting you say that because I've heard other people say that as well. It's not so much about the environment being there already, but you can complement to creating that environment for yourself,
I
in
think
which you're
that's
able,
key. Yes, I agree. I agree with you.
yeah, in which you're able to thrive, scientifically and intellectually. I heard someone say, you can almost somewhat bend the environment to your will or complement it, change it.
Well, you can
So
create if you have a big effect on the environment by the way you treat people and the way you act as a role model or lack thereof.
that's true. That's very true. Your actions do count. So, um, in terms of you being, in terms of you maintaining view of the bigger picture in your career and in your life in general, how have you done that? When you face challenges or obstacles, how have you maintained a view of the bigger picture that kept you going and that kept you persistent? How did you do that?
Well, I guess that's just me. I'm a very stubborn person. If I believe in something, I just keep trying and trying. I don't give up easily. If I feel something can do some good, if I feel that we've made a finding that's important, I'll do the best I can.
Okay, yeah, that's very good. So, Dr. Langer, I, I my career path. I'll just speak on my career path thus far. Um, I started off in engineering at Georgia Tech, and some things happened, and I transitioned to another institute, and I switched my major to biochemistry. So I changed path because also my interest changed as well. So my question to you is, for those in the field of engineering, what would be some of your advice, or what would be the key things that you did that complemented to you being able to transition to eventually a doctoral program in engineering and eventually to serve as a pioneer in the field? What were some of the key things that you did in your time as an undergraduate and as a graduate student?
Yeah, well, I think as an undergraduate, I feel you really want to learn the fundamentals. You want to learn the fundamentals and you want to really do well in class. I mean, that should be your highest priority. I mean, I wouldn't do research or other things until I knew I could do well in class, so to speak. As a graduate student, I think then, you also want to do the same thing, continue learning the fundamentals well, but then you really want to pick a research project and a research advisor if you're getting a PhD or even a master's degree who's really a good mentor. And what does that mean? I mean, hopefully that they treat you well and that they put you in an environment or have you in an environment where you can learn from them and from others.
Okay. Yeah, that's good. So I think most of us can agree that this time has been somewhat challenging for a lot of people due to the pandemic. And a lot of people have encountered obstacles or challenges. So if what has kept you optimistic during this time, during this pandemic? What has kept
Well,
you optimistic?
two things. One of the things I've been doing is I've been spending a lot of time trying to fight this pandemic. One of the companies I helped start's Moderna. Moderna has been one of the leading companies in terms of creating the COVID vaccine. So they're now in what's called phase 3 trials for doing it. And actually, almost every one of the companies that's doing an RNA vaccine, and those are the leading ones in terms of moving forward in the clinic, uses nanoparticles, which is an area to deliver the RNA. And, so that's an area that we've done a lot on. And we're also doing work in the lab on vaccines with masks, creating new masks, creating new ventilators, creating split ventilators, working on nasal sprays that might be able to combat it, working on new blood tests. So we've been very active in terms of really trying to hopefully make a difference in treating COVID. In addition to, even though research slowed down on other things because of the rules at the universities, I think that we continue that research too. And we're working a lot with the Gates Foundation on things that can help the developing world and just other things that I hope will be important to the world at some point. And again, I believe in all these things, so we're not going to stop, but all that is what we've tried to do.
So two more questions. In terms of your work with the COVID-19 pandemic, in terms of all these things you're designing and engineering, have you seen the impact, or is it still in the works? Have you started to see impact from your work thus far?
I'd say the answer to that is we've seen some impact. I mean, so what can I say? The fact that, some of the things that we did actually now over 45 years ago, creating, small particles that could deliver nucleic acids. Like I say, that's being used by Moderna, Pfizer, many companies. Again, lots of other people contributed too, but we did that early. We also developed other kinds of nanoparticles. And again, a lot of it's being done by our students and some of these places. So I think that the vaccines, they haven't made an impact yet, but other than the people that have been treated by it, but that seems to me the nation's biggest hope, the world's biggest hope is the vaccines. Other things we've done have had an impact. We helped make new masks, and several million of those have already been used by people, and I think they're better masks. And some of the others are in the works, but are in clinical trials, and they've moved very fast. So I think that we've been able to make some impact on patients already, and hopefully more.
Okay, so Dr. Langer, you mentioned, just going back, you mentioned how you said you're somewhat stubborn and you're persistent. Is it something that was taught you in your upbringing, or is it something you developed over time in academic settings?
I think it's something that I just was born that way. I think I've always been stubborn. I'm sure my parents thought that.
Okay. Yeah, well, Dr. Langer, it was so good to have you on today. I really appreciate you taking this time to chat with me and for us to discuss your career path and how you're currently making a difference and how you have made a substantial difference in chemical engineering and biomolecular engineering fields. So thank you again for joining me today.
Thank you. It's a pleasure and good luck.
If you haven't heard about Anchor, it's the easiest way to make a podcast. Let me explain. It's free. There's creation tools that allow you to record and edit your podcast right from your phone or computer. Anchor will distribute your podcast for you so it can be heard on Spotify, Apple Podcasts, and many more. You can make money from your podcast with no minimum listenership. It's everything you need to make a podcast in one place. Download the free Anchor app or go to anchor.fm to get started. Thanks for listening. We're glad you were able to tune into this podcast. Once again, this is The New Chemist, where we discuss chemistry, which simply put is the science of change, as well as the other sciences, careers, community, research, and COVID-19. Thanks again for listening. Note, the views on this podcast represent those of my guests and I. Dr. Langer's influential work has garnered him over 220 major awards. Among these are both the United States National Medal of Science and the United States National Medal of Technology and Innovation. His contributions to the field have been acknowledged through a vast array of other awards, including the Gardner Foundational Award, the Charles Stark Draper Prize, often likened to the Nobel Prize for Engineers, the Millennium Prize, the Priestley Medal, the Wolf Prize in Chemistry, and the Kyoto Prize. In 2015, Dr. Langer was the recipient of the prestigious Queen Elizabeth Prize for Engineering. Dr. Langer's research is at the cutting edge of biotechnology and materials science, focusing on the development of polymers for the controlled delivery of drugs, as well as the engineering of new tissues. His academic achievements are matched by his role as a member of multiple national science academies and his receipt of over 30 honorary doctorates from top-tier institutions worldwide. Very accomplished engineer with over 400,000 citations. His influence extends beyond academia into the realms of biotechnology and global health. Forbes and BioWorld have highlighted him as one of the most pivotal figures in biotechnology, and Time Magazine and CNN have recognized him as one of the most significant people in American science and medicine. It is definitely a treat to have Bob with us today, and it's good to have others joining in this informal mentorship meeting. Ugo, it's good to see you. So, drug delivery. We're just going to go through a few basic ideas in this presentation, and we will proceed to discuss some that's associated with some of the working systems. Drug delivery systems, according to the National Institute of Biomedical Engineering and Bioengineering, describe technologies that carry drugs into or throughout the body. So if we do like a quick overview, we have gone from ancient remedies and herbal medicines through the pharmaceutical revolution to oral dosage forms, to transdermal patches and biotechnology and gene therapy, to targeted drug delivery and smart drug delivery systems. So you have targeted drug delivery, biocompatibility and safety, controlled release systems, nanotechnology and nanomedicine, and personalized medicine, which is a really hot topic now. So some of Dr. Langer's achievements: Bob has achieved a number of things. Some of his work has been centered around investigating the mechanisms of release from polymeric delivery systems with concomitant microstructural analysis and mathematical modeling, studying and applications of these systems, including the development of effective long-term delivery systems for insulin, anti-cancer drugs, growth factors, gene therapy agents, and vaccines, developing controlled release systems that can be magnetically ultrasonically or enzymatically triggered to increase rates and angiogenesis inhibition. It's done a lot of work. So some of the big themes that kind of encapsulate the work that he's done: polymeric delivery systems, controlled release systems, angiogenesis inhibition, biodegradable polymeric delivery systems, tissue engineering and biomaterials, and some key questions. So this is going to be a dialogue. So some key questions that we have for you, Bob, are: how did the work that you did help with the research you're doing now in drug delivery? Once again, it's good to have everyone on.
Yeah. Well, so really, the way I got started in drug delivery was by doing cancer research, trying to have a delivery system that I could use to deliver large molecules, which hadn't been done before, from tiny particles. So that, and of course, at the time I did this was first published in Nature in 1976. I don't think there were very many large molecules that were of interest. Today, of course, there are many, and drug delivery has been key to that. I mean, not only for delivering, proteins, but, the recent work that one of the companies, as some of you know, that I started was Moderna, but the idea that you could deliver messenger RNA if you didn't have the mRNA, which is a large molecule, and nanoparticles, we wouldn't have the COVID vaccine, and I believe many other treatments that will come in the future. And of course, that is some of the things that we're doing today in drug delivery. We're working on delivering complex molecules by many routes: oral, through swallowable, through nanoparticles, through other routes as well.
Yeah, so we see that it advanced the field in certain areas. However, what were the lasting impacts made during the 70s, 80s, and 90s in your experience and opinion? During your early years of this work, what would you say were the lasting impacts that you saw then and that you are seeing now?
Well, I think the first impact was that you could do it at all. A lot of people didn't think you could deliver large molecules. In fact, the work we did when we wrote grants was rejected many times, and a lot of people who were experts just said that this was impossible, like trying to get a large molecule out of a tiny particle. They said either it would come out instantaneously or it wouldn't do it at all. And so, I think that was really the first thing. But then work by us and also by others, would lead to many things that I do think have had an impact. First, it's basic science. Trying to understand the mechanisms by which you can get delivery, trying to create new materials, trying to get the right kind of kinetics, different types of design. So many different things were done in the 70s, 80s, and 90s. And of course, I was fortunate. I had a lot of very great students and postdoctoral fellows who are now like professors or involved in companies all over the world. And, they did a lot of that work. And then there were lots of other people that I knew that was, got involved in this as well.
Okay, sounds good. So just before we proceed, I'll open the floor for any questions. Any questions that people have, I'll open the floor for questions, and then we'll proceed with the presentation.
Dr. Langer, I do have a question.
And you should just call me Bob. All of you should call me Bob.
All right. Wait, no, you know that I'm from the old school.
Well, you're an important person. You sir, all of you are.
Well, listen. What are your thoughts on now? You talked about nanoparticles, and typically the nanoparticles in the past were created using lyosomes, right?
Liposomes, yes.
Liposomes. Well, what are your thoughts on niosomes now?
Well, I think it's a good question. I mean, so people are making nanoparticles out of all kinds of things over the years. Whether it's lipids or polymers or really anything. I think all of them are interesting scientifically. I think the issues that I would think about would be, what I always call do they have some unfair advantage for a particular application compared to what already exists. And, you know, it might depend on the particular liposome or niosome or polymerisome or whatever it is that you have to do. So I don't know that there's a simple answer. And people may come up with ways of making them better and better. You know, interestingly, you know, when I started doing this, a lot of people also felt that lipid nanoparticles or liposomes were a nice academic thing, but they would never really be useful clinically. You know, and clearly that's changed because people figured out better and better ways to make them and better components. So, you know, and I think that's one of the great things about science. You can keep evolving it and making it better and better. So I always hesitate to say what's better. I think all these things are good, you know, and can have potential advantages.
Go ahead, Dr. Tim. Do you have anything else you want to ask?
No, we'll let that go.
Okay, yeah, that's good. Okay, so moving right along. So the presentation was composed based off of the extended list of articles that I found for that Dr. Langer played a part in writing or was an author in. So they were organized by years and segmented by each 100 in terms of how it was arranged in the list, and the themes highlighted that covered the ideas. So if you look at the early portion of Dr. Langer's, of Bob's work, we see that many principles, technologies, and concepts he developed during the 1970s and 1980s continue to be relevant and are actively applied in the design of modern drug delivery systems with a focus on improving therapeutic efficacy and patient outcomes. So some themes, so these are some from Bob's early work. So you had enzymatic generation of ATP in 1974 and 1976. Then we went on to the pH-dependent assays, mast cells and tumor angiogenesis, polymers for sustained release in 1976, the isolation of a cartilage fracture in 1976, and continuous long-term intraarterial infusion in 1978. Moving right along, his work then proceeded into the controlled release of macromolecules for polymers in 1980, zero water release kinetics in 1980, implantable drug delivery systems in 1981, microspares for controlled drug delivery in 1984, and local controlled release of diphosphonate in 1985. So, the question posed to Bob and you is, would you say that your work with Judah Folkman really laid the foundation for you to do this work that you did in the early portion of your career? Or would you say it was your graduate work and the work, your postdoctoral work with Judah Folkman that complemented to the progress, advancement of drug delivery research milestones during this time?
Yeah, I would say that it was definitely the work with Judah Folkman. I mean, he, you know, partly because of trying to solve the angiogenesis problem, and partly, you know, because, you know, and then the work with him, and then the later subsequent work at MIT that I did with different graduate students. But that was really the key things.
Okay, sounds good. Okay, moving right along. So the questions that we have: what were the foundational principles that guided your research inquiry during the 1970s and 1980s? What were like the overarching questions that you were trying to answer in the 1970s and 80s? And are they still influencing your current practice in the field?
Yeah, so I would say that, you know, part of it, it was trying to understand, you know, since people told us we couldn't deliver large molecules and I felt we were doing it, there were a couple of things. One was doing it and doing it very reproducibly. A second was really trying to understand the mechanisms. A third were developing newer and better materials. And I'd say some of those I feel we've solved, but they still influence what I do today. I mean, we continue to try to figure out, you know, how to make better systems and solve new generations of problems. I mean, you know, now that we have the foundation to make nanoparticles or microparticles or just deliver large molecules in general, you always want to do better, right? Can you target them to specific cells? can you cross barriers in the body that are tough to cross, like the intestine or the brain? But I think the foundational principles were.
But any thoughts on that, Aaron?
Yeah, absolutely. Well, one of the ones, I mean, there are other drugs like alchemies, which was based on some of our work they've been using. I think it's now naloxone or naltrexone. But you're right about buprenorphine. I mean, that right now is like in a sublingual system, and I believe some people are developing on injectable ones, like, and maybe that's what you meant, where they're sub-Q or intramuscular. I mean, I'm not an expert. I don't know what all the issues are. I mean, I think some of the things that I know people have talked about are, could you make them last longer? I think that's certainly possible. I know I talked once with Richard Pops, who was the CEO of Alchemies, which is one of the producers of the naltrexone one, and he said, that's a 1 month system. They'd like to make maybe someday a 3 month system or a 6 month system. So I think developing them longer probably would be, one of the things that would be useful. That particular system is called Vivitrol. But, and I would think that those kinds of principles could be extended to buprenorphine, but I'm not expert enough to know all the issues with buprenorphine.
Right. Vivitrol has worked well. I still think that there are some challenges with therapeutic concentration and maintaining that therapeutic concentration.
Making it flatter, you mean?
Yeah.
Yeah. Well, that's a good question, and that I think is something that could be done, by, I actually mentioned some ways long ago. I was actually on their board maybe 20 years ago, and I agree with, I think the ways to do it, you could, if you make sort of the effective particle size very, very tiny, I think you could probably help on that. And the other thing, though, maybe it's not a good idea from a manufacturer.
Okay, so it's really nice to have everyone gathered here today. My name is David Ferguson. We have Dr. Langer, who I'll introduce shortly, and my colleague and good friend, Quincy Britt. Would you like to introduce yourself, Quincy?
Hello, Dr. Langer. My name is Quincy Britt. I attend Howard University with David. And I'm also doing the Novartis internship this summer. So I'm very pleased and thankful that you've allowed me to come to this meeting with both of you.
Well, congratulations. It's nice to meet you. And people should call me Bob, so you don't have to.
Yeah, Bob. Yeah.
Okay. Yep. Well, nice to meet you, Bob.
Yeah, so just a quick introduction. Bob is one of the 9 institute professors at MIT. Being an institute professor is one of the highest honors that can be awarded to a faculty member. Bob has written more than 1,600 articles, issued over 1,500 patents, and is also the most cited engineer in history. He has served as a member of the US FDA's Science Board, the FDA's highest advisory board, and as its chairman. He has over 220 major awards, namely on some of them include the Queen Elizabeth Prize for Engineering, the Dixon Prize for Science, Heinz Awards for Technology. I had the privilege of attending his Kavli Award lecture, so he's also received the Kavli Prize, the Dreyfus Prize, the Medal of Science. He's a part of the National Academy of Medicine, National Academy of Engineering, and the National Academy of Sciences, and he also has been elected to the National Academy of Inventors. Bob is one of my mentors, and it's definitely a pleasure and an honor to be able to chat some more. So, Bob, just as we get started...
I saw the questions. I'm happy to try to answer them or answer anything I can.
Yeah, sure. So I'm about to raise the questions right now. So, in terms of throughout your career, you've emphasized interdisciplinary collaboration. How can emerging scientists strategically cultivate effective multidisciplinary partnerships early in their professional journeys?
I think it's part of working in an environment where that already exists, or working in an environment that is good for you, but where you'll also meet a lot of other people. I imagine actually working at Novartis, that's a good way to do it. But I also think, like today in academia, there are a lot of what I'll call interdisciplinary institutes. Like, you visited, David, our Koch Institute, which has not only biologists, but many engineers and MD. So I think that those are some of the ways.
Okay, sounds good. Sounds good. So, reflecting on your professional trajectory, what habits or mindsets do you believe were most crucial in transitioning successfully from scientific inquiry to impactful innovation?
Well, I think being willing to take risks
Okay.
and persistence, I think, are really important.
Yeah. Okay. Yeah. So, how have your mentorship strategies evolved over the years, especially when guiding individuals pursuing leadership roles at the intersection of academia and industry?
Well, I started out just doing academic work, but over the years, I wanted to see my work make a difference in people's lives. So I got involved in starting companies, and I also consulted for companies. You can only go so far in academics, and
Okay.
I felt like the companies, to me, were an avenue for, for doing things that I felt would help people around the world.
Yeah. So scalable impact. So, considering
David.
yeah.
Sarah, do you mind if I ask a quick question?
Go ahead. You're at liberty.
Yeah. So for someone who might want to start a company at some point in their career, what would your advice be?
Well, I think to get experience, maybe work at companies. I also think learning about patents is useful. But I think working at companies is really useful to get a feel. So I think like what you're doing is great. I don't know, summer you'll learn some stuff, but sometimes people might take a couple years and work at a company, and I think you learn a lot. But that's what I would think.
Gotcha. Thank you.
Okay. Sounds good. So, how do you foresee the integration of nanoscience with other disciplines like AI and bioinformatics in terms of reshaping pharmaceutical and biomedical research over the next decade?
I think what we'll see is artificial intelligence will be applied to all areas. Still the areas where where it can only make an impact is when you have a lot of data. But I think it'll be used in drug design, clinical trials, all kinds of things.
Okay. So could you highlight a recent breakthrough or approach in your lab related to nanoscience that significantly changed your thinking about future therapeutic possibilities?
Well, I'd say,
if you want.
Yeah. Well, no, I think our early studies many years ago that led to the discovery of angiogenesis inhibitors, that was important. But just knowing that you could create nanoparticles that could deliver drugs, I mean, that's helped on everything I've done from COVID vaccines to, angiogenesis inhibitors and so forth.
Okay, sounds good. So, just thinking back for a minute, when you developed those curricula for math and science education sometime back, what core principles guided your approach to ensuring both that there was the depth of content and also accessibility to diverse learners?
Well, I've always felt, and this probably will come up on an answer to your number of your questions, but I felt like on those issues, when I look at education, there's two things that aren't done that I tried to do that I felt were important. One was to make it fun. So sometimes I'd have games or things like that. The other was to make it relevant. So many things just seem so theoretical, but yet you can bring practical examples to people in science, like making rubber or even making ice cream. No, because ice cream you use freezing point lowering just as an example, because you can't make it cold enough otherwise. And then in math, I mean, there's lots of things. If you like baseball, I tell my kids and people like about batting averages and earned run averages. And then obviously taxes, the stock market, there's a lot of things where math comes in.
Okay, yeah, that sounds good. So, reflecting on your experience, what differentiates actually, considering current global challenges, how can aspiring scientists and educators ensure their contributions remain impactful, relevant, and ethically grounded?
Well, I mean, I think you have to try to do things that you feel are important.
Okay.
I think you always want to get advice about all those things, from your teachers and so forth. In terms of ethically grounded, I mean, I'd like to think that most things we do in research are ethically grounded, but if there's ever even the slightest doubt, it's good to talk to people that are more senior.
Yeah, I agree with you. So, finally, could you share a recent experience or insight that profoundly influenced your views on education, mentorship, or scientific innovation?
Well, really, I don't know if it was recent, but certainly I helping start the group school, which was a school I helped start with other people.
Mm-hmm.
It had a big impression on me in terms of the things I mentioned, that for education, you want to make things fun and relevant. In terms of research, I think my experiences early on with Judah Folkman, who was my postdoctoral mentor, who had really big ideas, that made a big impression on me.
Okay, so for as we start to wrap up, how did you balance the rigorous scientific accuracy with the practical engagement of students who might not naturally or initially be inclined towards STEM subjects? How did you balance that? So making sure you were technically precise when you were writing those curriculum, and making sure you were deep enough in terms of the scientific accuracy, but also making sure that it engaged the students well. How did you balance that?
Well, you do, and so you try to think of examples and problems that, depending on the stage of the person, that do that. And like I say, depending on where somebody is, it could be anything from sports to financial things, which are definitely relevant, and some of them can actually be fun.
Okay, so this is going to be the last question. Feel free, Quincy, if you want to chime in before you go. But this is going to be the last question. My question to you is, for people who are about to embark on nanoscience research or nanoscience studies or work in a lab that involves nanoparticles, what things should they look out for? What things should they really delve deep on? And where do you see the field of nanoscience heading in the next 10 or 20 years?
Well, I think nanoscience will head in a lot of different directions. In medicine, certainly drug delivery and imaging and diagnostics are important areas for it. But nanoparticles, I mean, they can be used in almost anything. And there are a whole class of materials. And I think, some of the places it'll go are probably straightforward, new vaccines, new therapeutics, so forth. But some of them, I don't think we're ever smart enough to know. I mean, people will make discoveries. I mean, if somebody asked somebody, 12 years ago what CRISPR was, or 13 years ago what CRISPR was, they would not have known. And I think it's been a great thing that people will continue to make great discoveries.
Okay. Well, Bob, thanks again, man. Thanks again. And this was a brief conversation, but it's very impactful as usual. So thank you again, and I wish you all the best, and I hope you all have a good rest of your day.
Well, you too, and it's very nice to see you again, and very nice to meet you.
Nice to meet you as well. Thank you.
Thank you.
Okay. Yeah, take care.
Welcome to the New Chemist podcast. We are so glad you are listening. Feel free to subscribe on Spotify and tell your friends and colleagues about the podcast.
Our Deepest Fear by Marianne Williamson. Our deepest fear is not that we are inadequate. Our deepest fear is that we are powerful beyond measure. It is our light, not our darkness, that most frightens us. We ask ourselves, who am I to be brilliant, gorgeous, talented, fabulous? Actually, who are you not to be? You are a child of God. Your playing small does not serve the world. There's nothing enlightening about shrinking so that other people won't feel insecure around you. We are all meant to shine as children do. We were born to make manifest the glory of God that is within us. It's not just in some of us. It's in everyone. And as we, our own light shine, we unconsciously give other people permission to do the same. As we're liberated from our own fear, our presence automatically liberates others. Our Deepest Fear by Marianne Williamson.
You are very important, especially to us here at the New Chemist podcasting group. You listening in is significant.
Vous êtes très important, surtout pour nous ici au New Chemist Podcasting Group. Votre écoute est significative. Usted es muy importante, especialmente para nosotros aquí en The New Chemist Podcasting Group. Usted escuchando es significativo.
Você é muito importante, especialmente para nós do The New Chemist Podcasting Group. Você ouvindo é significativo.
Είστε πολύ σημαντικοί, ειδικά για εμάς εδώ στο The New Chemist Podcasting Group. Το να ακούς είναι σημαντικό. Sie sind sehr wichtig, besonders für uns hier bei The New Chemist Podcasting Group. Es ist wichtig, dass du zuhörst. Je bent erg belangrijk, vooral voor ons hier bij The New Chemist Podcasting Group. Dat je meeluistert is veelbetekenend.
You are very important, especially to us here at the New Chemist Podcasting Group. You listening in is significant. Welcome to the New Chemist Podcast.
Bienvenidos al podcast del nuevo químico. Καλώς ήρθατε στο podcast του New Chemist. Welkom bij de podcast van The New Chemist. Bienvenue sur le podcast du nouveau chimiste.
Bem-vindo ao podcast do Novo Químico.
Welcome to the New Chemist Podcast. Work hard. Be value driven. You can do it. You can grow and learn it. You can be the difference you and your community needs. Don't give up. We are here rooting and cheering for you. Don't give up.
Travaillez dur. Soyez axé sur la valeur. Tu peux le faire. Vous pouvez grandir et l'apprendre. Vous pouvez être la différence dont vous et votre communauté avez besoin. N'abandonnez pas. Nous sommes ici pour vous encourager et vous encourager. N'abandonnez pas.
Trabalhar duro. Seja orientado por valores. Você consegue. Você pode crescer e aprender. Você pode ser a diferença que você e sua comunidade precisam. Não desista. Estamos aqui torcendo e torcendo por você. Não desista.
Δούλεψε σκληρά. Να οδηγείτε στην αξία. Μπορείς να το κάνεις. Μπορείτε να μεγαλώσετε και να το μάθετε. Μπορείτε να είστε η διαφορά που χρειάζεστε εσείς και η κοινότητά σας. Μην τα παρατάς. Είμαστε εδώ για να σας ζητοκραυγάσουμε. Μην τα παρατάς. Trabaja duro. Sea impulsado por el valor. Puedes hacerlo. Puedes crecer y aprenderlo. Usted puede ser la diferencia que usted y su comunidad necesitan. No te rindas, estamos aquí animándote y animándote. No te rindas. Werk hard. Wees waardegedreven. Je kunt het. Je kunt groeien en leren. U kunt het verschil zijn dat u en uw gemeenschap nodig hebben. Geef niet op. We zijn hier om voor je te roten en te juichen. Geef niet op.
Work hard. Be value driven. You can do it. You can grow and learn it. You can be the difference you and your community need. Don't give up. We are here rooting and cheering for you. Don't give up. Thanks for listening. We're glad you were able to tune into this podcast. Once again, this is The New Chemist, where we discuss chemistry, which simply put is the science of change, as well as the other sciences, careers, community, research, and COVID-19. Thanks again for listening. Note, the views on this podcast represent those of my guests and I.
Purpose of these episodes not at all for advice or medical suggestions. These are aimed to provide support to peer pharmacists in training in educational and intellectually stimulating ways. Again, these are not at all for medical advice or medical suggestions. Please see your local board and state-certified health professional. The views of this podcast represent those of my guests and I.
Welcome to the podcast where great minds meet. A podcast in which we host some of the most brilliant minds, science, engineering, and we've hosted the likes of Dr. Langer, MIT Institute Professor, Dr. Emily Brown, anesthesiologist statistician from Harvard Medical School, Dr. Todd Golub, Chief Scientific Officer at the Broad Institute. We've hosted the likes of ambassadors, engineers, professors, musicians, and so much more. It is such a treat today to have again with Dr. Robert Langer from the Koch Institute at MIT. Dr. Robert Langer is one of 9 institute professors at MIT. Being an institute professor is the highest honor that can be awarded to a faculty member. Dr. Langer has written more than 1,600 articles. He also has over 1,500 issued and pending patents worldwide. Dr. Langer's patents have been licensed or sub-licensed to over 400 pharmaceutical, chemical, biotechnology, and medical device companies. He's the most cited engineer in history. He served as a member of the United States FDA Science Board, the FDA's highest advisory board from 1995 to 2002, as its chairman from 1999 to 2002. He's received over 220 major awards. He's one of the 3 living individuals to have received both the United States Medal of Science and the United States Medal of Technology and Innovation. He has received also the 1996 Gardner Foundation International Award, 2002 Charles Stock Draper Prize, considered the equivalent of the Nobel Prize for Engineers, 2008 Millennium Prize, the world's largest technology prize, 2012 Priestley Medal, the highest award of the American Chemical Society, 2013 Wolf Prize in Chemistry, the 2014 Breakthrough Prize in Life Sciences and the 2014 Kyoto Prize. In 2015, Dr. Langer received the Queen Elizabeth Prize for Engineering. Among numerous awards, Dr. Langer has received the Dixon Prize for Science, the Heinz Award for Technology, Economy and Employment, the Harvey Prize, the John Fritz Award, General Motors Kettering Prize for Cancer Research, Dan David Prize in Material Science, the Albany Medical Center Prize in Medicine and Biomedical Research, the largest prize in the United States for medical research, induction into the National Inventors Hall of Fame, the Max Planck Research Award, the Prince of Asturias Award for Technical and Scientific Research, the Warren Alpert Foundation Prize, the Chirumo International Prize, the Benjamin Franklin Medal in Life Science, the Kabbalah Prize in Nanoscience and Nanomedicine, the Dreyfus Prize in Chemical Science, the Medical Science, the Medal of Science, the Maurice Chenot Award, the BBVA Foundation Frontiers of Knowledge Award in Biology and Biomedicine, the Bowson Prize for Biomaterials for Nanomedicine and Tissue Engineering. Most recently, he received the Dr. Paul Johnson Award for Biomedical Research and the Carvey Prize in Nanoscience. In 1998, he received the Lemelson MIT Prize, the world's largest prize for invention, for being one of history's most prolific inventors in medicine. In 1989, Dr. Langer was elected to the National Academy of Medicine. In 1992, he was elected to both the National Academy of Engineering and to the National Academy of Sciences. And in 2012, he was elected to the National Academy of Inventors. He has several other prizes, awards, and has received 43 honorary doctorates. It's definitely a treat and an honor to have Bob with us today. So the questions that we went through for this interview involve the following: determining strengths and mastering skills, dealing with failure and resilience, and serving others while in training. Some of the questions we discussed were: 1. How did you identify the specific skills and strengths that set you apart in the pharmaceutical and biotech industry, and how did you refine them over time? We also discussed what strategies have you used to accelerate mastery in complex scientific and business domains. In the constantly evolving, also we discussed in the constantly evolving industry, how do you determine which emerging skills or areas of expertise to focus on for long-term success? Also, we went on to discuss many breakthroughs in biotech come after setbacks. What is one of the biggest professional obstacles setbacks you face? And we discussed how do you personally reframe failure and rejection, especially when working on high-stakes pharmaceutical projects that impact millions of lives. As well as we discussed how did you balance rigorous training and career advancement with advice you give to someone early in their career as they seek to make meaningful contributions. So those are the questions we went through. Here are some of Bob's responses to those questions.
Yeah, well, I don't know that I had any intentional way of doing anything specific. I was looking really for a way to do things that I thought would help people with my chemical engineering background. But what got me into this area was working at Boston Children's Hospital as a postdoc in a surgery department. And it turned out that to solve the problem that we wanted to solve, which had to do with finding molecules that could stop blood vessels from growing, I had to develop a drug delivery system that could deliver large molecules. And that hadn't been done before, and that was considered actually impossible. But I spent a lot of just experimental time trying to do that. And eventually I was able to succeed. But then there were many, many other questions to answer. People didn't know how it worked, how to get the right kinetics, and all kinds of things like that. And so I
I just kept working and doing research and trying to answer questions with my students and postdocs over many years. I still am. Yes. Well, I suppose the earliest obstacles I encountered was, not getting any grants. And that was very frustrating. My first 9 grants were rejected. I just kept trying to do better, trying to understand what the reviewers wanted, doing more experiments. Eventually I did get the grants. And, and then I kept getting more grants, and so we were able to solve the problems. But that was one big issue. Another big issue was even getting a faculty position early on. The latter of the two is what you want to do. I mean, patent attorneys, you end up doing very broad work. I mean, it's not, what should I say? There may be end up being very specific areas that somebody might want to be expert in, but in general, patents, whether it's prosecution or litigation, I mean, there are all kinds of things, and of course, a lot of them haven't been even taught because they haven't been discovered or developed when you first started, education, or anybody else either. So I think a broad education is good, and I think pharmacy is certainly one of the areas that would be good.
Absolutely. Thank you, sir.
Yeah, well, I mean, different times in my life, it's been different. When I first started doing it, of course, we had no successes, and I just tried to be stubborn and push forward. Now we have had some successes, and I can tell my students, look, if you're able to solve these problems, it could save many, many lives. And I think, so I think people have to try to understand the positives that can come out of their research, and I think we can get that across.
Thank you. This is more of a general question that pertains to your journey. Was there ever a point, Dr. Langer, where you felt out of your depth, where you felt like, wow, like I don't feel good enough? And how did you overcome that?
Yeah, well, many times, probably still. But early on, I was doing this work in surgery and in, like I mentioned, the blood vessel work. And I didn't know biochemistry. I'd never had a biology course since 10th grade. That's still my last biology course. So I certainly felt out of my depth. I just kept trying. I'd talk to people. I'd read articles. I maybe understood 10%. I'd go and do some lab work. Maybe I'd get 10% right. I'd go back and read. But now maybe I could get 20% because I had learned some things. And it's through that process that eventually, I was able to do something.
Thank you.
Yeah, well, I think number one, you want to learn the fundamentals like chemistry, biology, physics, things like that. But beyond that, and then you want to learn how to solve problems, do experiments. And a PhD thesis in any area sort of tells you how to frame a question and then how to attack it and write it up and stuff like that. I think that's the way you do it, and then hopefully, you can learn from the literature, learn from the people around you, but you have enough skills that you can take advantage of what you know and read what you don't know and come up with ideas. I'm not really sure. I think it's just that I enjoy what I'm doing. I feel if I can help people, that's a good thing. So I keep trying and communicating that science can do good. That's something, I try to do.
Thank you. 2 questions here.
Well, thank you both, and I wish you both the best of luck. We'll be in touch. Thank you.
Thank you, sir.
Thank you. Bye-bye.
Thanks for listening. We're glad you were able to tune into this podcast. Once again, this is The New Chemist, where we discuss chemistry, which simply put is the science of change, as well as the other sciences, careers, community, research, and COVID-19. Thanks again for listening. Note, the views in this podcast represent those of my guests and I.
I'm fine. How about you?
I'm doing well. Thanks again for taking the time to chat with me. I appreciate it, man.
Sure, my pleasure.
So let's begin. So Bob Langer, Dr. Bob Langer is MIT Institute professor, Mosai Engineering History, FDA Science Board member, serving as chairman from 1999 to 2002, advising on the agency's highest level scientific matters, dual national medal recipient. He's also received the Charles Stock Draper Prize, the Millennium Prize, the Wolf Prize, among 220 major awards, the Lemelson MIT Prize, awarded the world's largest invention prize for being one of medicine's most prolific inventors in history. Recently received the 2026 Welsh Award, 46 honorary doctorates. He's a pioneer in drug delivery and tissue engineering, and he's an excellent, phenomenal mentor of mine. So it's definitely good to have him on today. Thank you, Bob. And this interview will be used in the newsletter. So I built this conversation around a question I genuinely don't have an answer to. Can multi-receptor pharmacology and smart drug delivery be unified, or are they separate paths to the same destination? Your work defined one of those paths, the glucose-responsive patch, the emergency glucagon implant. Each is an answer to a question like, how do we give a therapy the capacity it needs to act without the patient intervening or without the patient's input directly? So what I would like to do in the next few minutes is walk through both arcs, identify where we're beginning to touch, and then put 5 open problems to you that I generally value your perspective on. So the first question is, Bob, can multi-receptor pharmacology and smart delivery be unified, or are they best kept separate?
Yeah, I think they can be unified, but I don't think it's straightforward. I think it's, I think really the idea of, in general, a theragnostic, or a system that can diagnose things and tell you what to do, I think is a good idea. But, I don't think that it's not necessarily that simple. I always think it's good to be able to do one thing well first, but I do think it's certainly possible. You know, but it really hasn't been done in this case, but the answer to me would be theoretically yes.
Theoretically, yes. Okay, cool. I understand. So, what aspect of your work do you think leans closer to that, combining the two, or from what I've seen, is it best as your work really dealt with it separately?
Well, I think at a high level, you know, we've certainly done things where you can diagnose things and where you can deliver things. One of these things was like an implantable microchip that we developed with Michael Sema and John Santini, and a system like that, I think, does have the potential, you know, to put the things together in it that could do that. But that's from our work. There's certainly other people that have done work that might be relevant as well. But from our work, that kind of system, you know, I think could someday be useful.
Okay, that sounds good. So, in an incretin-dominated era, we have so much about GLP-1s. Does autonomous or smart insulin still have a defining role or a necessary role?
I think so. I think still. I mean, first, not everybody takes these substances, and secondly, I think we don't necessarily know, you know, the really long-range effects. And even if you did, I mean, I don't think diabetes is going away. I mean, I think that. So I think certainly insulin has a place, and I think smart insulin has a place.
Okay, yeah, I agree with you, especially for type 1 diabetes, yes.
Right, especially for that.
Yeah. So, moving right along, can delivery plus mRNA move metabolic disease from management towards modification or even potentially cure?
For diabetes, you're saying?
Yeah, for diabetes, yeah.
I think mRNA can be very, very useful. I think it depends on exactly how you're trying to use it. I mean, the beauty of mRNA is you can make anything with it. You can get any gain of function. So certainly mRNA could deliver, you know, it could be made to deliver insulin, it could be made to deliver glucagon. I think, but a cure, I don't know that that's necessarily a cure, but I think it certainly is something that could be useful.
Okay, yeah, I understand. So, given this, based off this question, where do you see the therapeutic modalities or therapeutics headed now for mRNA in terms of diabetes?
Well, I think, you know, I'm not sure how. I mean, I think different people are doing, you know, looking at different diseases for mRNA. I think diabetes is not at the top of the list for that, just for, you know, different reasons. I think mRNA, you know, for cancer is being widely studied, especially by Moderna, but by others as well. You know, that seems like there's clear breakthroughs there. And certainly for rare diseases, these kinds of things are very useful. But if there's a molecule you wanted to make, like glucagon or something like that, I think certainly this could do it. I could make insulin too. I mean, you can make anything you want with messenger RNA.
Yeah, I agree. Okay, so moving right along. So can the mRNA, LNP, and nanomedicine platforms move metabolism from management to modification? Do you think that's possible?
I do think it's possible. Yeah, I think it's certainly possible.
Okay.
It depends what you're doing exactly. It kind of relates to your last question.
Yeah, I agree. So as we start to bring this conversation to a conclusion, how do these advances reach a global scale, and how do they reach an equitable scale?
Well, I think that's a good question. I think, well, the first thing that I think is important to realize is that a lot of the issues on equity and global scale don't have to do with science. I mean, in other words, they have to do with transportation, they have to do sometimes with politics, they have to do with a lot of different things. In our own lab, we've done a lot of work with the Gates Foundation trying to address at least the scientific questions on that. Can you make shelf-stable, messenger RNA? Can you put it in patches that might be stable, regardless of the environment? Can you even hopefully make it, less expensively? But I don't know that solves the problem of a global scale. My understanding of the global scale issues, and we're involved even in that in nutrition, is there's a lot of different political issues, there's a lot of transportation issues, there's a lot of, logistical issues. And those, as far as I can see, whether we're doing nutrition help or whether we're doing messenger RNA help, I think are really the predominant issues, and they're not easy to solve. It's not something that's just science.
Yeah, I agree with you. Yeah, those issues are very complex and multifaceted, and they involve multiple partners and stakeholders. So, in terms of the work that you've done, have you done any work in terms of therapies that can increase adherence or compliance for patients?
Yeah, we've done a lot of work on that, and that's with the Gates Foundation has funded it for exactly that reason. Even though equitable things are very important, compliance, as you pointed out, is also very important. And if you go to Africa, and you look at the statistics from the World Health Organization, the percentage of people that don't take medications because of the issues you mentioned are much worse than in the developed world. So a couple of things we've done. With GeoTraverso, we've made pills that can last for the entire course of treatment. We could make them last for a month, for a week, maybe even a year. With anajacolenic in our lab, we've developed what we call self-boosting vaccines. So theoretically, you could just take one injection and you never have to come back for a second. And that's been a big issue. Now, it's important to realize that the pills we're doing are in clinical trials. The self-boosting vaccines, we actually have done polio in non-human primates, and it works well. But it's always a long road and very expensive to solve all these problems with the clinical trials. But we have done many things to help patient compliance, and clearly they do reduce it. With the Gates Foundation, for example, we're working on a once-a-month oral contraceptive. We're also working on a once every 2-week system to stop malaria in small communities. So there's that's been something of, and we've developed better, I think, children's medicines. There's terrible compliance for children, and with Dr. Traverso, we've developed a number of these, and they're in clinical trials right now.
Okay, Bob. Thanks again, man. I appreciate all this information. These are good nuggets, good insights for me, especially as I do my clinical rotation. So definitely applicable, definitely could add more perspective as you take care of patients. So me in the clinic and you in the lab. So definitely, it was good to chat with you, Bob, and I appreciate you taking the time to do this.
My pleasure.